Hi guys. For today, I just want to continue the the alignment that we couldn't finish it last week. And if you could get the files down, the analysis, that's good. And if you not, just copy the files from the directory that I provided in the course website, and then just have them in a directory in the in this directory that I'm going to go to it. Okay. Download the files inside the alignment output if you do not have the analysis done last week. And when you get the files, so you should have the BAM file and the sorted BAM file and the log file that just shows what's the results of BAM file. And in order to visualize these BAM files, this alignment, we need to first index this BAM file. The BAM files then should be downloaded at the same time with this index of BAM file, the reference genome, and the annotation of the reference genome. So to create the index for this BAM file, we again need to do a QSUB first. The last, okay. I think we can also work from waiting. I think I have those files already done. The index file, if it does not work, we can do another copying the index file of the BAM. I'm not sure.
Like some people are getting on. Your work.
Yeah, 8 people on the right now.
Yes. Everybody, if you're waiting on one and it does not take you to one, so connect with the time here tonight. On this device here, there are computers you need for. You don't think you need one, you get rid of yourself, I think. All right.
Do what now?
So if you don't put it in, people claim you get one of those computers yourself.
You get the entire day to yourself.
Which is a problem. And because you can equals one, that means you and me and him, or we can all be on the same computer. There's more jobs. If you don't put a PC in one, then they don't have 8 jobs. If you were not on, you would not have to get it. You're going to get to an exit. You need to hold on to it. You currently have a computer all to yourself.
So while I'm waiting, I just copy the index file of the BAM file. If somebody wants to copy it to the directory that the BAM file is existing, just use this path. This one, Luster, Haven courses, and the rest is here, alignment output, and then copy this one to your current directory. Did you log in or did you get it?
Anyone here still waiting? Can you raise your hand?
I think it's better just copy this index file because I think we need 2 other parts of the still remaining. Let me just check. No problem. Yeah, this one, the highlighted one. Yes, and this is a folder, this is a directory, and then copy this file. One, RNA-seq 1. That's BAI. Yep. Does it allow you to download? Does it allow to download the permission? Could you get it downloaded or? Okay. So just go ahead. If anybody got the file, just raise the hands or. Okay. Did you copy? Okay. No, no.
What I'm saying is some people not putting in PPA. So. Yeah. Then use your lights on 35.
So guys, for the students that got the files, just go to the directories that the files are aligned, and the alignment file and the index file are located. The one that is alignment output. And make sure that this span file and the index file that you just copy are available. Then copy the 4 files that I already mentioned: the index file, the bound file, the index of the bound file, the reference genome, and the annotation of the reference genome, which the location, the directory are of each of these 4 files located in the course website. And just instead of the user, put your username, net ID, which by the command.
Then use your install.
SCP. Download all the 4 files and put them in one folder.
Anybody else story? I'll down to the wrong side. Your story? I'm story too.
Okay, what's happening? Does anyone download the files to their local computer? You waiting? Okay.
Okay, there's the confusion that people don't know where the maps are, or you're confusion in getting to their password. Okay, so we've already heard from Vicky that you have to add the ATF. It's duo.tennessee.edu, and I think it's duo.tcf. Yeah, I think it's just a typo. The problem is that when I try to download that BAM file, it's always going to take 8 minutes. Is that about what everybody else is facing?
Yeah, the file size is more than 1 gigabyte. That's the BAM file.
So what's it? I mean, I guess you can leave that running in the background.
Okay.
And then people still waiting on their system to start.
But they can copy at least open another window and just copy the index file that I created into your directory that your BAM file exists. Then you can go ahead and copy those 4 files into your local computer.
Right, but we're gonna have, we also wait on files, yeah.
Okay.
Finally, 6 minutes, that's just over file. Did you use BAM, the BAI, the reference genome?
But
No, I wait on it.
Nick, do you think it's better to go ahead with HTTP? Start just explaining what the parameters are, or it's similar to last sentence?
Like 30 to 40 minutes left. And we can just try to wait for. Unfortunately, mine's getting like slower and slower. It's not telling me like 12 minutes. It's kind of like, I think we're all down.
So because for the HTC, again, we need, at least for the basics, we need that QSOF, and then we need for a whole processor, for a whole node, you have to wait 14 minutes. We do not need to wait unless the students want to have their analysis and their outset done. They can copy the results again. But for the HTC, I can just talk about the parameters. Okay. Okay, guys, while we're waiting for this multiple things that is happening, for the lab 2, for the RNA-seq, to get these, after aligning and getting this the mapping matrices, we need to identify the to count the reads, and then put those reads inside a statistical package called, in this case, DC, and then get the differential expressions. So in this case, we have to get our interactive sessions done first, then use the HTC to count the number of the reads that have been mapped to the reference genome. So in here, where is it? Okay. The software HTC needs 2 required arguments, which is the alignment file, which is a BAM or SAM file, and also the annotation file. And before these 2 parameters, we need to have the options, which could be, in our case, that I'm talking about this now, will be format, which is exactly specified which format you're using, BAM or SAM. The order, which means that you are using the alignment files that have been assorted based on the position, and this stranded one, which shows that whether the assay that you are using is strand specific or not, which in this case is no for our analysis. The type, which is different between the different, actually it's 3 different types in the parameter type, which is exon, transcript, and gene. In our case, it will be gene. And the last one is just identify when you are adding the numbers to the genes, which parameter, which attribution of those annotation is needed. And in this case is gene ID, which exactly we got it from the annotation file. If we got those interactive sessions, if we could get them done, we need to first load the software, the HTC. I think mine works. But before loading the software, we need to create some folders and directories for our current analysis. So just go to the course website, to the course directory, to your username. And then create the Is anybody still waiting for getting the QSOF? So, in the rest of the website, the course website, the first code relate to a specific, a single one per gene that you can run it, which it takes about 35 to 55 minutes per one whole node. And the second batch of code is for example, you have 13 paired end reads to analyze. So instead of doing one by one, we need a for loop. The second one actually is for a for loop. It's a for loop command to analyze multiple files at the same time. And since we need these files, the output. Yeah. For this band file.
Do we want to work on as a group?
Yes,
Okay, let's do it.
sure. Okay. So we need to just inside the current directory that you are, just create a folder, a qsub command called, let's say, http and then you start typing. Yes. I need that one. Okay.
Skip the queue. Skip that line. Hey Nick, if we don't specify the debug queue, it should still work, right?
Yes, only on the opportunistic. On there. I would go ahead and try it anytime.
Try the debug queue? Or try the institutional one? Okay.
So this is perfect. This is like the bare minimum script to get you started on launching something on the ACF without needing an interactive session, which is great. We've talked about lots of PDF options, but this is sort of the minimum you can get away with. Don't forget the CD, TVSO worker. It's usually a good idea to just put it at the top, make sure you're in the right directory, or in the directory you think you're in.
And then after loading the software, you just need to copy and paste the code that I've already mentioned in the course website after the module load HTC after the loading the software, which is
It's very subtle, but it's a dash L, a space, and then a node equals 1. So L 1. Very hard to. No, I think we're not. I think this is just going to be faster in the future. You can do this while you're on the login node, right? Like, or this is really what the debug queue exists for, right? Is so that you can get onto the APF and start installing your script. So if you do this on the login node, if you're not having one, that's fine. Because all you're doing is taking into the text editor. And then we can stop it. The system will take care of putting this on the appropriate computational resources. You won't have to worry about it. It'll be much faster. Yes, login say yes and do it from the login.
okay. All the parameters the same as the website. Just copy it in here. And then save it. And then make it executable.
Hey Nick, is it not going to say the typical thing?
I wrote it.
Okay. But I like it because I like to know if I wrote the script to go into view or if I wrote the script to have on the on. I probably shouldn't teach people that. Not say this, but.
Do you need the code again?
Yeah, where on the website.
Yes, it's on the course website. Just add it after the module load HTC. Yeah, it's in the RNC tab too.
I think we're inside
HTC RNC Lab 2 and then HTC, and make sure that you're in HTC while you are running this command or submit it to the ACF.
That was all okay.
It's all lowercase HTC. Then just submit the job by the command qsub htccount.qsh and then enter, and then you will have a number that has been assigned to your command and job. That if you want to check where it is, you can check qstat, which is on Q, the second one. The first one is actually the one that you asked for an interactive session, and the second one is actually the foreign job that you just submitted. And I think it should be like this. Okay. Did you get the files download it into your directory, local computer. You got it?
Well, there's a band file.
Okay.
Like,
Everything is slow. Try and check it.
So for example, under satisfying how Q, this is Q. And then change it to R.
Okay, so I found the issues. We need to add one more thing. So everybody can still wait, but maybe do it. And then hit the up arrow to the same line, bring it up again. So what you're gonna do is after the colon EPN equals 1, do a comma, and then do end access policy.
Yes.
My Meyer.
Yep. You can type it here.
Okay, I can type it there. It's fine. So after the, so that's the very last thing in your line right there. So after you're going to do that, add that at the end of PBN equals 1. And then give it about 30 seconds, and you should get in.
Is this for the rest of the semester?
Do what?
Is it useful for the rest of the semester?
It shouldn't be. So we just pushed out a new submit filter last week. They made a bunch of new changes to it. We're still finding bugs in the submit filter. So the way it's set up now is it's set up where one user can submit as many jobs as they want with PPN equals 1, but multiple users cannot submit multiple jobs with PPN equals 1. That's why there's only 8 people in the queue and everybody else is waiting. So this should be a temp fix. For now, yes. Yes. So there's a bug, which is the reason why we had to put this at the end of the line. Once we fix the submit filter, you shouldn't have to put this, but As long as you put ppn equals 1 at the end, you should be able to get on every time. But I'm not sure how long it's going to take the admins to fix. So you should be able to get on every time, hopefully. If you have that at the end. Yeah, I just got on. So it takes about 30 seconds. So don't control C it or anything. Yes, everybody is at that. At the end of the end. Yeah.
This one is only for the yeah. You can also add this one to your queue sub for the the first line. So it goes like this. Notes 1. We didn't add the rest in the queue sub. You can add the rest.
Yeah.
If we were to use those, do you want us to back out if we do it this way? Okay.
We wait to see if anybody else is already doing it. You might know.
And then the, anybody want help? Oh, you think, okay.
Make did you get the file?
And the final, we'll just wait for it to set up and now it's very.
Okay, so I think we can go. But is anybody around to waiting for the files to be downloaded to his or her local computer? Did you get it all? Okay. So, yes, 16 files. Actually, just one by one, I run them, and then you have to copy those 16 text files to your local computer. It's in the next steps of this time.
Oh, okay, so your.
Which one? The files to download?
No, I got the file downloaded and my surgery to work on.
Oh.
Interactive, it's done, so, yeah, it's fine. Okay. I'm able to load Java running on my
Also, did you download the Mac version? I think you have to uninstall it and reinstall it. So if you have already downloaded the IGV, open it please, and then load the genome annotation first, the genome reference genome first. And then go to tools. No, let me. After uploading the genome reference, Load the genome annotation and the BAM file. The BAM file. And to get more information about the annotation, the splice junctions and this kind of stuff, go to tools. The second option, run IGB tools. Change the command from count to sort. Select your annotation file, which is this one. Then run it. Takes a couple of seconds. Okay. Then load the annotation. What's going on? Seems something is wrong in my file. So after you upload those 4 files inside the IGB, you will be able to visualize the alignment map and the annotation on the bottom part. So let me just again do it the same. This annotate the reference genome. Then the annotation. when you upload the when you upload the files into IGB, you have to choose the chromosomes that the reference genome does have. Choose one of them, let's say chromosome number 5, and then you will see if you zoom in to the files, you can see the alignment for each read. For example, in this case, in here, this is the annotation on the bottom line, and these are the alignment, and the upper one is the reference genome. Does anybody face a problem? Each section? Okay, sure. First, from the genome, load genome from file. Then go to the folder that you have your reference genome downloaded, and select the one that ends with dna.genome. This is your genome file, reference genome, actually. Then after you opened it, just close it, and then from file, load from file. Again, it pops up this window that your files are there. Then you choose the GTF file, and then open for another window. Again, we will load the BAM file, which ends with .out. Yep, yeah, it doesn't matter where you put them. Just make sure that they're all in one folder. .genome.fa. Yes. .genome.fa. Ready to do stuff? Where are you? Yeah. Just close it, reopen it. Because
Yeah, anything I do is something
so maybe the file, you have to only upload the BAM file. That's BAM, not the BAM.bai. Yeah, I think let me. Okay, wait. Let me. I think the files have a problem. Maybe the original file. Let me double check it. Can you check what's the size of the file that you download to your local directory? Okay. Are they exactly enough to this sizes? It should be in this directory. It seems that the BAM file for everybody has a problem. The BAM file. And I think maybe yeah, it also worked for me, but I don't know, maybe during transferring it has a problem, but I think everybody has a problem in the uploading the BAM file. So maybe I just remove all the folder contains the BAM file. I will overwrite on it, and you can just copy it again and rerun it, this IGB. Sure. Just one thing. Since the main problem is from the SAM file, the BAM file, I replaced it with the current BAM file. I just remove it and replace a new one with the one in the website. But follow the course website and check out this video to how upload the reference genome, the annotation, the BAM file, and then you will get this information by zooming in into each alignment file.
Yeah, try to use the print in the file. That's what I really need to do. Hold on. Keep an eye on that, and when you come back on this day, hopefully we'll have file. And try to get it working on it, but don't. It seems like it's not ready for the computer, so I'm not gonna worry about it. I'm not that. It's nice to have, but I have no idea how to troubleshoot it. Maybe a long time.
Thank you, guys.